Okay, last video on cell membranes. This video will help you to answer number 1. Can you give examples of membrane-bound organelles in eukaryotes, describing both structure and function for each? And can you explain why compartmentalization in eukaryotic cells is helpful in maintaining homeostasis? So this is our endomembrane system, which is basically a series of membranes that are in the cytoplasm of a cell. These membranes help divide the cell into different compartments. So what we're looking at here is our Golgi apparatus, our smooth endoplasmic reticulum, or smooth ER, and our rough endoplasmic reticulum, rough ER, and the nucleus. By having all of these different membranes, we help compartmentalize the different processes that are occurring in the cell. We'll talk about that in a minute. But we're also, by having this addition of all the membranes, increasing the surface area, which helps to allow for more space for whatever it is to be. So for example, in our rough ER, we have all of this extra membrane, and that extra membrane creates more space for the ribosomes to be. The more ribosomes, the quicker the process can occur. Same thing happens here in our Golgi apparatus. The more membrane we have, the more vesicles we're able to make to increase the speed of transportation of particles in and out of the cell. Having all of these membranes helps to localize metabolic processes that occur within the cell. So instead of saying, for example, photosynthesis just occurs in the cell, we can determine that photosynthesis occurs in the chloroplast in the cell. So specific processes occur in specific places, as opposed to it just being a big mess. Some other examples of this would be in the mitochondria, where we know cellular respiration occurs, and the nuclear envelope and endoplasmic reticulum. So we're now going to go piece by piece of all the things that have membranes in the cell. Right here we have our nucleus. The nucleus is made up of several pieces. The first thing is that there's a nuclear envelope that surrounds it. Obviously, remember, our nucleus holds genetic information, genetic material. The nuclear envelope has several pores throughout that allow for the movement of particles in and out of the nucleus. And our centerpiece, often represented by a darker dot, is our nucleolus. Next, we have our smooth endoplasmic reticulum, which is located just outside the nuclear envelope. The role of the smooth ER is to modify proteins for cellular functions and structures. The rough ER looks very similar, however the rough ER has ribosomes attached to the surface. The role of the rough ER is to package proteins for secretion. Here's an image of our nuclear envelope and the rough and smooth ER. All right, our next organelle we want to talk about is our mitochondria, which are present in all eukaryotes and are responsible for the production of ATP. That process we know is called cellular respiration. All right, and then we have our chloroplasts. The chloroplasts are present in photoautotrophs, so things that can take light and turn that into energy or into sugars, and it is the site of photosynthesis. Now, in comparison to our eukaryotic cells, our prokaryotic cells, so our archaea and bacteria, usually lack internal membranes and organelles. In addition, they have a cell wall that surrounds them. Their structure is going to be much simpler than eukaryotes because they developed before eukaryotic cells did. And what these cells have is enzymes embedded in their membrane, so you can see it against this top image here, and that's where their reactions are going to take place. So in prokaryotic cells, enzymes are embedded in the membrane of the entire cell. That's where all of their metabolic processes are going to occur. Whereas in a eukaryotic cell, we have all of these different organelles where our processes will occur, and this is a great image for you. So going back to those original questions, examples of membrane-bound organelles in eukaryotes, describing both structure and function, We went through that. And compartmentalization is key to maintaining homeostasis because we are able to separate out all of the different processes into specific locations, allowing for more efficiency. More efficiency makes it much easier to maintain internal conditions of the cell than as if it were all combined.